So let me talk to you today about how we use a little bit about how vestibular hair cells in our inner ear work, and with the theme being how they're specialized to work fast, because we use them. Oh dear, how do I advance? There we go. We use them to, one of the things we use them for is to compensate for our own rapid head motions, allowing us to stabilize gaze and balance. Uh, so they need to work fast, and you'll see that in amniotes we have some specializations for that. So just along the fast theme, and to reset us a little bit, I'm sorry, I don't know what you've seen already, but the fastest reflexes we have are the vestibulo-ocular reflexes, and this is just to set you up for that. The best studied one is the angular or horizontal vestibulo-ocular reflex, and it involves 5 synapses from hair cells through the nerve, brain stem, and onto the motor neurons that drive muscles that control eye position. And it's the reflex that allows you to maintain gaze when your own head is moving. When you rotate counterclockwise, the reflex rotates your eyes clockwise to stabilize gaze. And this is a remarkably fast reflex, as it needs to be to give you online stability as you're moving. The data here are from the Cullen Laboratory, and they show the eye velocity in response to a head velocity transient, so a sharp head motion delivered to a large-headed primate. You can see that the delay is just a few milliseconds. And so it's in order to drive those reflexes that we need really fast reception and transmission in the inner ear. All right, so the outline of my talk is, we've already done part 1. I'm going to introduce the mouse utricle. It's our model preparation, but I'm very glad that it's a model for many other ways of studying the inner ear in mammals. For obvious genetic reasons. In our case, we chose it partly because it has advantages for stimulating and recording once it's prepared in a dish. But we really like the fact that we know so much about this preparation from the work of others. I'm going to talk a bit about how sensory information is organized on the vestibular epithelium through zones that give rise to different afferents, through directionality, expression by the hair bundles. And I'm going to talk about those 2 hair cell types that are quite famous and belong only to amniote vestibular organs. And I'll sort of conclude with their sort of salient specializations, which involve potassium channels in the hair cells, also elsewhere, but we'll focus on the hair cells, and specialized synaptic transmission. So I like this picture from David Lim of the saccule. It's a sister otolith organ of the utricle, so they're both linear accelerometers and can also detect head tilt. The utricle is especially good at that. So tilt is, a change in the head position relative to gravity, and that cannot be distinguished within the ear from linear acceleration, so it's always present. So here we have, the reason I'm showing the saccule is that this shows a nice cutaway of the structures. You can see it's a layered structure with the otoconia that move relative to the underlying epithelium when you undergo linear acceleration or head tilt. And the otoconia are coupled to the sensory epithelium through a gel layer, which contacts, in some cases, or just floats above, in other cases, the hair bundles of the sensory epithelium. And that below that epithelium houses the hair cell bodies, and the synapses, which give rise to afferents, which project to the brainstem and cerebellum, depending on which zone they're from. And you can see this really remarkable difference in the central stripe, which is the name it's given. It's called a streola, which just means stripe. And I'm pointing it out because it will show up throughout the talk as a zone that has highly specialized hair cells and afferents, and you can see accessory structures. It's meant to do a different kind of job than the extra streola. All right, so here's our utricular macula, the sensory epithelium of the utricle. This is the preparation we work with. Typically, we excise the epithelium. We have to remove those lovely otoconia because we need access to the hair bundles and hair cells for stimulation and recording. And we usually keep the nerve and cell bodies in order to preserve the synapses within the epithelium. And here you see a couple of canal cristae that usually come along for the ride and are part of our entire preparation. If we look inside the epithelium, say right here, that little dotted line, we see the beautiful hair cells. I have to admit, these are not properly attributed. These are utricular hair cells, but they're from the cat utricle, and they're from Hunter Duvar. And they show it quite beautifully the classic shapes of two types of hair cells that we find in mammalian and other amniote vestibular inner ears. Here's the supporting cell. So let's look a little bit more closely at these. The most prominent feature of hair cells was, can everybody hear me? Okay, good. All right, so you're also quiet. No, I'm just kidding. The hair cells were first really noticed, I think, because of their the amazing synapses that you find on this hair cell type 1. So if we just look at them over here, they have different shapes, but the really prominent feature, and functionally one of the most important features, is that the afferent forms a cup-shaped postsynaptic terminal that envelops the basolateral membrane of the hair cell. And whereas the type 2 hair cell, which is more like other hair cells and other organs, receives Bouton contacts from often the same afferents, which are opposed to presynaptic ribbons. So we have this extraordinary postsynaptic calyx contact. It's the only postsynaptic calyx terminal or cup-shaped terminal that we know of. Oh, there they are. Great. They're actually there. And now you can't doze off. All right. So, well, there's no grade. So the hair cells of just a little evolution, how do we get these unusual hair cells? The hair cells of anamniotes are similar to type 2s or vice versa in having these kind of Bouton contacts and in other features. But type 1 hair cells are shaped by all amniotes, suggesting they emerged after tetrapods moved from water to land about 300 million years ago. And that's, I don't know the exact timing, but at about the same time, we evolved cochleas. So for this, especially to be able to pick up air-driven sound. So something about that transition had powerful effects on both compartments of the inner ear. And so amniotes share the type 1 hair cells. And here I just show that amniotes all have flexible necks that allow them to have independent motion of their heads, may create more vibrations. They're moving on land often, which is a really hard surface and may produce a lot of vibrations and possibly be much more difficult to navigate than water. All right, so just quickly, you can use your hair cells, the disposition of hair cell epithelia by itself sort of helps provide information about head direction. So like I mentioned, the vertical and horizontal otolith organs, obviously the canal organs in different planes. And hair cells only detect motion of their hair bundles, the component of motion that is parallel to the plane of the epithelium. So that's one way in which information is conveyed. Then within the otolith organs alone, there's a variation in hair bundle orientation. So each hair cell has a preferred orientation. And again, it only really picks up the component of the bundle deflection that is in line with that orientation. So this variation in hair bundle polarity is sort of follows a line of polarity reversal, which runs through the middle of the approximate middle of the epithelium. Here you see it from the side with bundles facing each other. At each point along that curved line, bundles face each other. So you have a slow progression in orientation. And I don't have time to show you, but it's always fascinated me how variable the actual orientations are compared to what they show, what we all show on these schematics. But in some sense, that's related to the directional sensitivity of hair cells, which is not that sensitive to deviations within about 20 degrees of the preferred angle. As shown a long time ago with careful mapping of how the response varied with direction of the stimulation relative to the hair bundle. So maybe it doesn't matter that much. It's only as good as it needs to be. And this is the sort of average polarity. All right, and this is just to show you what we see from above, where you can see the orientation of bundles. This may be a bit small for you guys, but if you look, we're sort of near the line of polarity reversal, which meanders through this zone right here and along the whole epithelium. And I just hope you can see that the bundles are sort of wedge-shaped with a kinocilium at one edge, sort of the tall edge of the bundle. The hair cell responds to motion in that direction. And so these hair bundles on opposite sides have opposing responses to a given head motion. Okay, the bundles, if you moved so that the bundles moved in this direction for this side, that would excite them and it would inhibit the others. All right, so associated with that line of polarity reversal, but not in an obligatory way. We now know that different genes control the line of polarity reversal. And we also know that it has a sort of different relationship with the sensory zones within the epithelium in different species. So it's not obligatory, but they do both sort of crawl through the middle of the epithelium. And here you see a schematic of zones, the sensory zones, I call them, because they are associated with really different sensory properties. So the strialar zone here in orange and here, and the extrastrialar in blue. And as I mentioned earlier, the afferents from those zones are really different from each other. Their spike timing is different. The strialar zones have highly irregular and extrastrialar highly regular timing. And that is interesting in its own right, because they're sort of extremes of the two different ways you can present spike timing, but also perhaps functionally it's very important because it's correlated with and at least in part causal with differences in the response dynamics of the two sets of afferents. So the strialar afferents are more adapting, more sensitive to motion transients. For example, if you've just learned about vestibular evoked potentials, they're responsible largely for those because those are transient motions. They use both temporal and rate codes if you're a neuroscientist, and they directly project to the brainstem, putting them in a good position to drive those brainstem reflexes that I briefly alluded to that allow you to respond rapidly to your own motion. In the extrastriola, you have really extraordinarily regular firing. As a result, they use just a rate code, just change the interval. They're tonic, non-adapting, carry the head tilt information and low frequency. They're more sensitive at low frequencies. And some of them project directly, the lateral ones here project directly to the cerebellum, where they provide input to sort of calibrating information where, for example, adjusting reflex for head tilt, or adjusting the response to a linear translation depending on the influence of gravity, which is also affecting the sensory epithelium. And some of them, the MES afferents actually project to the vestibulo-ocular reflex pathway. All right, sorry, that was lateral and medial extrastriola. All right, are there any questions? Just taking a breath. All right, let me switch to how we stimulate and record from hair cells. Yes, was there something?
I think you have to turn on your microphone. Oh yeah, we're good. Thank you.
We're good? Yeah.
Yeah.
All right, so I'm going to start with recordings from single hair cells. If I have time, I will briefly talk about some new work we're doing. I probably won't have time, but I always throw it in. All right, so what I told you that this is our preparation, and here we're looking down on a little zone of it, little region of it, interest, right down on the hair bundles as we see them experimentally. And we're applying hair bundle deflections to move the hair bundle along its preferred polarity or orientation. So here you see it the way we see it in experiments. So we bring in a glass rod, for example, and couple it to the bundle and move the bundle back and forth. Are you guys seeing my cursor?
Yes.
Yes, okay, good. I could use the highlighter if I could figure that out. No, I apparently can't. All right, so we'll just use the cursor. So moving the hair bundle back and forth along its preferred axis. So we're getting the maximal response with this stimulus. And we bring in below the surface, we bring in a pipette sort of laterally to approach the basolateral membrane of the same cell, and apply suction to form a tight or gigome seal, which allows us to then basically suck a hole in the membrane, but it's tight, the pipette is tightly sealed, so there isn't a leak of the electrical signal or ions out of the cell, except into the recording pipette. So all the current that the cell produces now goes up the pipette, and we have access to it for recording. So this is whole cell patch clamp recording, and it's really revolutionized over decades our ability to collect really lovely data from small cells like hair cells. And with this kind of method, we can study the transduction current, which is generated in the hair bundle, and also the currents through channels in the basolateral membrane of the cell, this part down here. So transduction channels are up here. Those are the ones that are gated by the hair bundle deflection, and the other channels are in the cell membrane. So basically, the cell puts as many transduction channels as it can in the bundle and doesn't waste any territory up there for voltage gated channels, which are down here. And there are a lot of those. So during this voltage clamp recording, voltage is not allowed to change. The only thing that's changing is the position of the hair bundle. So this just shows it in 4 different positions and the current that you record through the transduction channels as a result. So it's generated by just maybe 100 channels. There's a small number of channels, a couple maybe per stereocilium. They're typically 50 to 100 stereocilia. So it's a small current. And here it is for step stimuli, and you see this prominent adaptation of the response, which we don't have time to talk about, sadly. But we can also see that in the sinusoidal waveforms, which are sort of a favorite in my laboratory. It allows us to kind of get just an overview of the tuning, frequency tuning of the cell. And what I'll show you here is that now we're oscillating the bundle back and forth like this with a sinusoidal waveform at different frequencies. And as the frequency rises from 2 hertz to 100 hertz, the response gets bigger. That's a manifestation of this adaptation. So another way to put the adaptation is that the response is bigger for a fast transient stimulus than for a sustained stimulus. And that's what you're also seeing with the sinusoids. I hope that's clear. All right, so then what happens? So here, let's just walk through the basic stages that we can access electrophysiologically with patch clamp recordings. So we're going to move this hair bundle back and forth sinusoidally. That's the stimulus again. And it goes from 2 hertz to 100 hertz. And now we're going to start with a recording from the hair cell in voltage clamp. So there we're just looking at the currents. And in current clamp, where we're looking at the receptor potential, which is the cell's more natural signal. And it's influenced by the voltage-gated channels in the cell membrane down here. You can see that it has a really different shape from the transduction channels, current. All right, so there's a lot of filtering being done by the hair cells, basolateral membrane, its channels, and also its passive properties. It takes time for this current to change the voltage of the cell membrane, and we see that in the fall off of the responses at high frequencies. And then if we then jump the synapse and record postsynaptically for the same stimulus to the hair bundle, we can see really radically different looking responses. And these are quantally transmitted postsynaptic currents and postsynaptic potentials. So those are mediated by transmission of glutamate from vesicles at the presynaptic ribbons. And you can see now that the response is very distorted compared to the original stimulus. It's got a sort of peak shaped, it's no longer really even quasi-sinusoidal. And finally, from the same location, we can see spiking, which takes place down here at the heminode, or the node where there's a big concentration of spike generating channels. And so I just want you to see how all of these stages are a sort of cascade of processing that takes you from this shape, mediated by the accessory structures, to this signal, which is very much tuned around the intermediate frequencies. That's for a type 2 like hair cell, even though I showed it on a type 1, which is kind of let's call it a baby type 1 can behave like that. In fact, that's where the data were collected, which is why I'm showing it. Baby type 1 hair cells electrically look a lot like type 2 hair cells until they grow their calyx more and acquire a whole lot of potassium channels. Let me just briefly show you the thing we're trying to do now. So many people have recorded 1 unit at a time from afferents across mammalian utricles and other vestibular organs, and compiled a sort of view of how the average striolar afferent responds and the average extrastriolar. But we don't actually know how, and the same thing goes for the hair cells. We don't actually know much about how those properties are distributed moment by moment across the epithelium. And that seems really important because sort of in the last decade, it's become clear that you need, and partly from a beautiful paper from the Lorenz laboratory, showing that you need, a hefty collection of hair cells to serve vestibular function. So if you can regenerate 20% of the hair cells, that's not good enough. You get no recovery of function that's detectable. So it seems that you need to have a big, robust, coordinated signal coming into the brain from the sensory epithelia. And you have to recapitulate that through regeneration or repair mechanisms or vestibular implants. And so we're kind of interested in understanding more about how hair specific head motions are represented across the two-dimensional surface of the sensory epithelium. And to do that, we started to do calcium signaling, which has some known disadvantages. It's slower than electrical signaling, but we are calibrating it against electrical signals, which I don't have time to show you. I just wanted to give you a feeling of what it's good at, which is allowing you to see how a common stimulus co-activates a bunch of cells. And in this case, we're going to look at the specific calcium signal produced by this hair bundle, which belongs to a type 1 hair cell. And I'll see if this whole thing will work. Well, let me try again. Yeah, they're going together now. All right, so this is the bundle of interest, and this is the bundle of interest, and this is the calcium signal produced by this motion in microns in black. The stimulus delivered by a fluid jet over here is shown in gray. And what I'm just showing one response, and apparently among our many technical difficulties, it's not showing the full trace, but maybe too big a file for transatlantic transmission. Anyhow, so, but you can see that if I just run it again, you look here, you see all of these bundles lighting up for that common stimulus. And we are taking these co-activated signals and measuring their coherence and other properties to sort of try to understand the common signal that's sent for a given type of head motion, which we can control with the fluid jet. We can approximate with the fluid jet. So trying to get beyond single cells and think about each, afferent in most cases innervates more than one hair cell, multiple hair cells, and in most cases, at least in mammals, they're mostly both type 1 and type 2 hair cells. So we want to kind of understand the input to a given afferent. We also can look at the calcium signals of the afferents. All right, enough of that. Okay, so let me conclude with some discussion of why we have two vestibular hair cell types in amniotes, which really I'm going to truncate as why do we have type 1 hair cells, since in a way all hair cell organs have type 2-like hair cells. And type 1 cell, I'm only going to focus on the ways in which all type 1 cells differ from type 2 cells. So although we're working in the mouse utricle, we think that these observations apply to Christie and non-mammals. Like there's evidence that it applies from work from other people, important work. For example, the first patch recordings from type 1 hair cells were done by Marie Skłodowska-Curie's group in pigeon. So a lot of important comparative work I'm just not touching on, I'm afraid. All right, so what we can say is that the hair cell type has a loose association to functional zones. So there are we can say that type 2 cells are everywhere in the epithelium. Type 1 cells are always in the striola, and in mammals, they're also in the extrastriola, and this is also true in the Christie for the central and peripheral zones. The ratio of type 1 to type 2 varies across species and zones. But in our preparation, it's actually close to 1-to-1 in both these striola and extrastriola. So there are cool specializations in the striola, like complex calyces that envelop more than one type 1 hair cell, but there are sort of similar numbers of type 2 and type 1 cells across zones. So the reason that's important is that it's been hard to sort out what type 1 cells bring to the picture since they're everywhere. And the main functional differences we know about, or functional specializations we know about, are zonal. So it just has been hard to parse that. But we can say now that what we can say is that type 1 hair cells have unconventional voltage activated potassium channels that shape the receptor potential unconventionally. And that maybe the main purpose of this effect is to shape a unique form of afferent transmission that, as far as we know, only occurs from type 1 cells to calyx afferents. And it depends on the calyx being there as well. All right, so in hair cells in general, potassium channels are kind of deployed. There are large numbers of different kinds of potassium channels, and hair cells always have potassium channels make up the dominant type of ion channel in the basolateral membrane, and play the largest role in shaping the electrical properties of that membrane, which means it shapes how fast a voltage can be created from an incoming transduction current, and also affects the signal, of course, that's delivered to the next cell, and so on down the line. So we have these specialized potassium channels in the hair cells at the postsynaptic membrane, also in type 1s relative to type 2s, and also in striolar afferents relative to extrastriolar afferents. There's different ion channels at the spike initiation zone. So I'm just going to talk briefly. How am I doing? I have no idea when I started. Let's see. So let me just run you through what's that?
I was just going to say, yeah, we have, if you start wrapping up, then we'll make sure we have some time also for some questions, but I think
okay.
That's accounting for a little bit of the delay we had. So, but take your time, and we wanted to make sure we have some time for questions. So thanks.
Okay, thanks. So just to really, because I didn't want to not show you the bewildering complexity of things, but let me walk you through what happens to remind you. So a hair bundle is deflected, and positive ions enter the apical hair bundle tips through the transduction channels up there. And in vivo, those ions are potassium, and there's an important component of calcium as well, right? But there's high potassium in endolymph, and as a result, the main ion coming in is potassium during that positive deflection. And during a negative deflection, you actually reduce the influx of positive ions. So there's a standing entry of positive ions. And then that entry of positive ions depolarizes the cell membrane, both cell membranes, and leads to efflux of potassium out the base, where there's a reverse gradient. So potassium leaves the cell. So it's a very potassium efficient mechanism where it comes in at the top and leaves at the bottom without requiring pumping to sustain the gradients. And I've thrown in some efferent channels and some calcium channels. The calcium channels in hair cells are absolutely critical for mediating glutamate transmission, vesicular or quantal transmission. And this is over here. So we see potassium channels, calcium channels, postsynaptic glutamate receptors. And then we have efferent channels on efferent terminals landing on the calyx and making contact with different kinds of of acetylcholine receptors, which also interact with potassium channels. So we now know that both hair cells in the mouse utricle express large numbers of a sort of weirdly overlooked kind of potassium channel called KV1.8. It belongs to the big family of Shaker channels, if you know anything about potassium channel lineage. So it's in a distinguished family, but it's been overlooked. We didn't pay much attention to it because heterologously, that is when expressed in other cell types, these cloned channels have very positive activation ranges. And the one thing we knew about the type 1 hair cell potassium conductance was that it activated at a really negative potential, well, negative to resting potential. So it was weird in that way. And this wasn't shared, as far as we knew, by the channel. But what Hannah Martin, working with me, found was that both the dominant channels of type 1 hair cells and the quite different behaving dominant channels of type 2 cells depend on the same subunit. So that's the beauty of potassium channels. They can do different things in different cells, even the same subunits. And I just want to use this slide to show you that when we look at a null animal, we see a big effect of removing the potassium channel from both hair cell types on the speed of their response. So buried in here in black is the stimulus delivered by a probe to the hair bundle. So remember, moving the hair bundle towards its tall edge evokes it evokes a current in blue that is, oh no, this is voltage, sorry, receptor potential in blue. It has to be voltage because it's depolarizing. And it's very fast in the type 1 cell and pretty fast in the type 2 cell, but not startlingly fast. So you can hardly see it in the type 1 cell because it rises so quickly. So that means you deflect the hair bundle and the hair cell produces a receptor potential almost instantly compared to the type 2 cell. And other hair cells are not this fast either. Well, if you remove that subunit, then that effect goes away and you get a more conventional, slower rise time of the receptor potential. In both of them, it looks rather similar. Okay, so takeaways, they both need KV 1.8 subunits to have a fast receptor potential, but the type 1's is really fast. But the cost it pays for that speed is size. So the type 1 hair cell, here we're looking at responses, receptor potentials to the same frequency series that I've been using. And you see that the type 1 cell has like broad tuning. It doesn't get smaller with increased frequency. That's because it's fast, but it's really small, right? It's just a few millivolts. And a type 2 hair cell, for comparison, can at low frequencies has a gigantic response, tens of millivolts. And it just doesn't do as good a job at holding that amplitude as you go to high frequencies because for one reason, it's the nature of the KV 1.8 expressing channels is different. And that is that the channels in the type 1 cell are open at rest. And so the cell has, it's electrically leaky. It has a low input resistance in both, and that has both effects, speeding up the response, which is proportional to the resistance. So the time it takes for the transduction current to charge the membrane is proportional to resistance. And the size of the response, V or delta V, is also proportional to the resistance. So the cost of speed is gain in this system. And that's true throughout the nervous system. These kinds of channels are not these specific ones, but channels that are more open at rest are used throughout the nervous system to make responses faster. But there's a cost in sensitivity or gain. So just to quickly show the big difference in the open probability of the channel in the type 1 cell, it's fully open at minus 60 millivolts. Minus 75 might be its resting potential. The type 2 hair cells channels, also KV 1.8 channels, are not open until you get above the resting potential. And so that's the difference in input resistance. So why have so many potassium channels just to make the receptor potential small? Well, as we discussed, you make it fast. Maybe that's the most important thing. But also these channels contribute to a direct form of transmission that bypasses quantal transmission and has, as a result, a very fast component, a very fast capacity. So quantal transmission, I don't have time to walk you through it, except that it's a cascade of events. The receptor potential changes the open probability of calcium channels, which promote in a various charming dance of molecules, the fusion of vesicles with the presynaptic membrane, dumping glutamate onto the postsynaptic membrane, activating channels. So it's like all these stages to get to a spike. Now, what the transmission that I'm talking about that only type 1 cells do goes straight from receptor potential to postsynaptic potentials very rapidly. Okay, so this is, I wanted to say, this is what quanta produce. So an excitatory postsynaptic potential produced by quantal transmission has a skewed shape. And we actually saw some of those earlier. So it's these are distinctive features of quantal transmission. And the difference between type 1s and type 2s was first noticed a long time ago by a great physiologist, Haru Omori, and his colleague Yamashita, who showed that if they recorded from afferents innervating chick type 2 hair cells, they saw the expected EPSPs, sort of the currency of the nervous system, everybody thought. But if they recorded from calyx afferents, they saw something that looked like a slightly filtered version of the hair cell receptor potential. These are very high frequency stimuli, 250 hertz, a sort of vibrating ball above the bundles. And this amazing result has finally been duplicated in numerous ways. It lay fallow for about a decade because people couldn't believe it, that there was this weird kind of transmission that wasn't an artifact and didn't seem to depend on quanta. So this transmission, why have it? Well, I just want to illustrate its nice features here, where we're recording from one calyx that's getting quantal transmission from an immature hair cell, type 1 hair cell, and non-quantal transmission from a more mature type 1 hair cell at that stage of maturation in the first postnatal week. And you can see that the non-quantal response, it's less rectifying. It has a both positive and negative component. So it's a more faithful representation of the hair bundle stimulus. And it also shows less tuning. It's sort of more broadband, especially out to higher frequencies. So non-quantal transmission is faster and more linear, which might be really helpful for vestibular reflexes, where you're trying to drive muscles to do the opposite, for example, of what your head is doing. So you want a really linear and fast response. And this transmission has been shown by our colleagues at Rice University, Rob, Raphael, and Aravind Govindaraju, to, especially Aravind, this is his thesis work, to emerge from what we know about the ion channels pre and postsynaptically, and from the calyx. So putting those two things together, these specialized potassium channels in the hair cell, they're complementary but different channels in the postsynaptic membrane. Putting these together with the calyx, you get sizable non-quantal transmission, actually physiological transmission. And I'm about to conclude. That is illustrated here. If we just look here at the postsynaptic response, simulated postsynaptic response to a 1 micron hair bundle deflection towards its tall edge, the response has, in red, has two components that can be analyzed because it's a simulation. It's a really nice feature of the model is that you can analyze what's going on. The fast component in blue combines with a slow component. So the fast component is an electrical change in the synaptic cleft due to the hair bundle motion, causing a sort of instantaneous electrical change postsynaptically. And then the slow response is due to the gradual accumulation of potassium. Remember, potassium comes in and exits, and then it's kind of trapped there by the calyx, which seems to be kind of on purpose because it really amplifies the signal and gives you, in this case, a spike, which doesn't occur with either of these components alone. All right. So, so with these simple properties, the calyx and the, the previous slide was just suggesting through simulations that how you could get this non-quantal transmission. If speed is what you're evolving towards, then you make synapses bigger. And if they get big enough, they start to accumulate potassium, which is generally considered to be a bug, not a feature, but it seems to have been run with by these type 1 hair cells and taken advantage of. And there's evidence now that this non-quantal transmission is sufficient to produce a lot of the behaviors that we study that depend on type 1 cells. It doesn't mean that the quantal transmission is not doing anything, but it's possible it's not doing too much in the mouse. So just to sum up, type 1 cell, why do we have type 1 cells? They evolved as animals moved onto land, and it's a much more challenging environment than water, as Chris Platt, a beloved scientific officer in the United States, once said to me, it's hard to trip in water. So you need your type 1 hair cells to drive reflexes that allow you to compensate for your own tripping and prevent you from falling on land, which is a more treacherous environment than water in some ways. And with that, I will conclude. Thanks for your attention.